Think of a situation where your teacher distributed a specific circular among all students in your class. She might have taken the Xerox of the original circular to make the desired number of copies, right? Likewise, you might have taken copies of your birthday invitee to distribute them among your friends. Can you think of any situation in which cells make copies of certain molecules or organelles? When a cell divides, copies of its organelles are formed, which are then distributed among the daughter cells, right? Now, think about the DNA molecule present in each cell of the body of all living beings. When the new cells are formed, the existing DNA molecule should make copies, which are then distributed among the newly formed cells. It ensures that the newly formed cells have the same genetic information as those present in the original parent cell. But can you tell how the copies of the existing DNA molecules are formed? Does this process require any specific enzyme? What is the part of the eukaryotic cells where the process occurs? Do the prokaryotes such as bacteria also copy their DNA in the same part of their cells? The process of generation of copies of the existing DNA molecule is called DNA replication or simply replication. Let's discuss the process in detail to find out the answers to the above questions. Recall the double helical structure of DNA as explained by Watson and Crick. Accordingly, a DNA molecule is composed of two complementary strands of opposite polarity. They propose that the complementary nature of two strands of a DNA molecule should allow each parental strand to serve as a template. But how? The two strands of a DNA molecule should separate to specify the nucleotide sequence of the new DNA strand. If the parental DNA strand has nucleotide sequence AT, it will specify the synthesis of the DNA fragment with TA sequence. That is complementary base pairs. This is because adenine in the parental strand would pair with thymine and thymine in the parental strand would pair with adenine. Once the synthesis of the new DNA strand is completed, the complementary bases of the parental template strand and the newly formed strand should make hydrogen bonds. Therefore, the resulting DNA molecule has one parental template strand and one newly formed strand. The parental strands are shown in black and the newly formed strand are in grey. Each DNA molecule formed by the DNA replication has one black colored parental strand and one grey colored new strand. This model of DNA replication in which each DNA molecule has one parental strand and one new strand is called semiconservative replication. I have a doubt here. How do we know that DNA replication is a semi-conservative process? Was there any experimental proof provided for this scheme? The experimental proof of semi-conservative DNA replication was first given by Matthew Michelson and Franklin Stahl in the year 1958 in the bacterium Escherichia coli. Let's discuss their experiment in detail. Nitrogen present in the bases of DNA occurs in both heavy, that is N15, and a light, that is N14-4. Michelson and Stahl grew Escherichia coli cells in the presence of N15 for many generations. It produces E. coli cells having all the DNA heavy labeled. At the start of the experiment termed generation 0, they switched the bacterium to a medium that contained only N14 and the DNA double helices were collected after 20 minutes, 40 minutes and 60 minutes, represented as replication cycles 1, 2 and 3 respectively. This is because The doubling time for E. coli under the growth conditions provided by them was 20 minutes. Since the medium contained only N14, all the newly synthesized DNA strands were labeled with light nitrogen. However, the original strands would remain in heavy form. Michelson and Stahl then analyzed the density of the DNA by cesium chloride gradient centrifugation. The DNA molecule with both strands having N14 has light density and remains around the top of the tube. A DNA molecule with one strand having N14 and the other strand having N15 is said to have intermediate density. The presence of N15 in both strands makes the DNA molecule heavy and moves it closer to the bottom of the centrifuge. So, can you predict the results? Here, after one generation, two DNA molecules of intermediate density were formed. After 2 generations, the mixture of DNA with light and intermediate density DNA was observed. This is because the two parental heavy density DNA strands shown in dark color were present in 2 DNA molecules which are labeled as 2 and 4 here. The rest 2 DNA molecules labeled 1 and 3 consisted of newly formed light density DNA strands only. The newly formed DNA strands are shown in blue color. So, the results were consistent with semi-conservative replication. I have a doubt here. Are there any other possible modes of replication? Yes, these are conservative and dispersive modes of DNA replication. The parent strands form the double helix after DNA replication, while the two newly formed strands base pairs to make a double helix. So, the original parental strands are conserved. If DNA replication was conservative, out of a total of 4 DNA molecules, one would have a heavy density, while the rest of the 3 would have a light density. This is clearly seen in this image. However, the sedimentation of all DNA molecules as a single band disproved the formation of 2 distinct types of DNA molecules by the conservative model. According to the dispersive model, the segments of parental and newly formed DNA are interspersed in both strands after replication. The dispersive model predicts the DNA of intermediate density after 1 generation and after 2 generations. Each DNA strand should have 1/4 heavy nitrogen and 3/4 light nitrogen. But the results were not consistent with the dispersive model as well. Taylor and colleagues in 1958 used radioactive thymidine and observed its presence in the newly formed DNA in Vicia faba. The results of their experiments were also consistent with the semi-conservative DNA replication. All these experiments proved the semi-conservative nature of DNA replication. You might ask a question here. What are the enzymes involved in the process of DNA replication? The process of DNA replication includes many enzymes, but here we will discuss only some of them. Let's begin the discussion with DNA polymerase. As the name suggests, the DNA polymerases are the enzymes that polymerase the deoxyribonucleotides. It means that these enzymes catalyze the formation of phosphodiester bonds between the deoxyribonucleotides to make the long polynucleotide chain of DNA. DNA polymerases exhibit certain features. Let us check them one by one. These enzymes require a template and add the deoxyribonucleotides according to the nucleotide sequence of the template strand only. So, if the template strand has AAAA, the DNA polymerases would catalyze the formation of the DNA strand with TTTT sequence. Another feature is that DNA polymerases cannot begin the synthesis of new DNA strand de novo and require a DNA or RNA strand with a free 3' OH end. The DNA or RNA fragment with a free 3' OH end is called primer. Generally, the primase enzyme synthesizes RNA primer, which is then elongated by DNA polymerases. Since DNA polymerases add new deoxyribonucleotides to the free 3' OH end of the primer or the growing end, the replication of DNA occurs in 5' to 3' direction only. This means that the incoming deoxyribonucleotide added to the 3' OH end and extends the DNA strand in 5' to 3' direction. Do you know that E. coli has at least 3 types of DNA polymerases called DNA polymerase 1, 2 and 3? Among these, DNA polymerase 3 is the main enzyme of DNA synthesis. Likewise, human cells have at least 5 different types of DNA polymerases. Despite the differences, all these enzymes have 5' to 3' polymerization activity. This means that these enzymes add deoxyribonucleotides to the free 3' end of the primer or the growing end of the DNA strand. Did you notice? If DNA polymerases catalyze the synthesis of DNA strands in 5' to 3' direction, can 2 strands of DNA with opposite polarity be replicated in the same direction? It means that the template strand with polarity 3' to 5' is replicated continuously in 5' to 3' direction by DNA polymerase. But what about the template strand with 5' to 3' polarity? The problem is solved by discontinuous replication of the template with polarity 5' to 3'. It includes the repeated formation of primers at small distances. These primers are then extended by DNA polymerase in a 5' to 3' direction to form the short DNA fragments called Okazaki fragments. These Okazaki fragments have RNA primer at the 5' end, which is then removed, and the gap between the successive Okazaki fragments is filled by the addition of deoxyribonucleotides by DNA polymerase 1. You might ask a question here. How are these Okazaki fragments joined to form the continuous DNA strand? The answer is DNA ligase enzyme, which uses the energy of ATP hydrolysis and joins the successive Okazaki fragments. So, the replication of the template with polarity 5' to 3' occurs in a discontinuous manner, while the template with polarity 3' to 5' is replicated continuously. Is the parental DNA unwound for its complete length to serve as a template? The answer is no. Breaking down the hydrogen bonds between the complementary base pairs to separate the 2 DNA strands and then prevent their pairing are energy expensive processes. So, the 2 DNA strands are separated in a small region to serve as a template, and this region is called replication fork. The Y-shaped structure in this image is a replication fork. The template strand with the polarity 3' to 5' is replicated in the direction of the movement of the replication fork. The resultant daughter DNA strand is called the leading strand. On the other hand, the template with polarity 5' to 3' is replicated in the opposite direction of the movement of the replication fork, so that the DNA polymerase can make the new strand in the 5' to 3' direction. The resultant daughter DNA strand is called the lagging strand. I have a doubt here. Where does the replication of DNA begin? Is there any particular site or point, or does it begin at any random point? The process of DNA replication begins at a specific site called the origin of replication or ORI. ORI is required for the replication of DNA, and this is why the introduction of DNA fragments into the host cell and its replication requires ORI, which is then provided by the vector DNA. We will not discuss the vectors here. You might ask a question here. When does the DNA replication occurs? Recall the phases of the eukaryotic cell cycle. The S phase of the interphase of the eukaryotic cell cycle includes the replication of DNA. Once DNA replication has occurred, the cell must divide into daughter cells. If it does not occur, the cell would not have the doubled DNA content. The presence of more than 2 copies of complete genome is called polyploidy. So, the absence of cell division after DNA replication gives rise to polyploids. Now that you have learned about the process of DNA replication and the associated machinery, can you tell the enzyme responsible for the formation of RNA? What is the process of synthesis of a new RNA strand called? When and where does it occur? Like DNA replication, does the synthesis of RNA also require a template strand? If yes, what serves as a template? We will discuss the process of RNA synthesis in the next video to find out the answers. Now, let us summarize what we have learned.
Manorama Horizon. Learning made simple.